Hi. So today I'll be talking about the role of reinforcement in error-based processes on exploratory motor behavior, particularly in neurologically intact and Parkinson's disease. So exploration is critical for discovering new and successful actions. In humans, we commonly see this when learning a new sport, like basketball. But exploration can also play an important role in neurological rehabilitation, where we need to push patients outside their comfort zone a bit to find a globally optimal solution that helps their problems. Now, one way we can observe exploration is through statistical random walks. And one study done in 2013 looked at statistical random walk exploration in humans in a visual motor reaching task. Specifically, they looked at exploration along task redundant dimensions. Here, I'm showing one of the targets from their experiments. The black dots represent the final reach positions participants saw at the end of each trial during the task. The researchers broke this task down along task relevant dimensions along the minor axis of this target and the task redundant dimension on the major axis of the target. To quantify exploration, they used a lag-1 autocorrelation analysis independently on the task relevant and task redundant dimensions, where they found statistically greater exploration through lag-1 autocorrelation on the task redundant dimension. And the researchers attributed this to the presence of error-based corrections in the task relevant dimension, reducing the exploratory behavior. Recently, our group, through a series of 3 experiments using binary reinforcement feedback, show evidence to suggest that this exploratory process can be regulated by a reinforcement-based process. Now, where do these 2 systems live in the brain? Correction due to visual stimuli has been attributed to the cerebellum, whereas modulation of behavior in response to reinforcement feedback has been attributed to the basal ganglia and the dopaminergic system as a whole. Both of these systems have been shown to independently influence exploration, but there is evidence to suggest that they can interact in a unique way in sensory motor exploration, as they share interconnections with one another, as well as shared indirect connections with the premotor and prefrontal cortices. Now, one powerful way we can gain causal insight into how one of these systems influence exploration is to observe a population with a deficit to one of the systems. And as such, we recruited people with Parkinson's disease who have a deficit to the basal ganglia and dopaminergic system to gain causal insight into the role of reinforcement-based processes specifically in exploration. We used a stochastic model-based framework to encapsulate our hypothesis and drive our predictions, and it uses movement variability as a natural way to explore a task. Most notably, our model incorporates terms that incorporate reinforcement-based processes in the light blue, and the light orange are terms that are associated with corrective error-based processes. So for experiment 1, we hypothesized that error-based and reinforcement-based processes are dissociable and interact in sensory motor exploration. And specifically using our model, we predicted that reinforcement-based processes boost exploration, while error-based processes suppress exploration. So in experiment 1, participants were seated at a kin-arm robot, and they were told to reach and stop inside of a target they saw on a screen. The white dot represents the start position, and the long rectangle is a target that they were reaching to. We had 3 conditions, and in the first condition, participants received reinforcement feedback at the end of the reach if they landed inside of the target. So when they stopped inside the target, it would expand and glow blue, they'd hear a pleasant noise, and receive a bit of money towards performance bonus. In the second condition, participants received visual error-based feedback, meaning wherever they stopped, they would see a little cursor appear to say that's the point that they reached to. And in a third condition, participants received both forms of feedback at once. So wherever they stopped, they would see a cursor, but if they were inside of the target, it'll expand and glow blue, they would hear a pleasant noise and receive a bit of money. Here, I'm showing our general model, simulating the final hand positions of one individual running through both our reinforcement and error feedback conditions. And one thing you can note from these final hand positions along the target is that there's much more spread along the major axis with reinforcement compared to error feedback. We can quantify exploration using lag-1 autocorrelation analysis along the major axis of the target, where our model would suggest we should see the greatest level of exploration with reinforced feedback. More importantly, we can do this exact same analysis in a representative subject, and we see our model predictions in our data, showing that we should see greater exploration with the reinforcement feedback condition. So here I'm simulating 36 individuals running through our experiment. On the x-axis, we have our conditions, and on the y-axis is our lag-1 autocorrelation analysis. What our model suggests is that we should see the greatest level of exploration with reinforcement feedback the lowest level of error feedback, and intermediate levels of exploration when given both simultaneously. And more importantly, we see our model predictions again in our data. We found statistically significant differences between all three conditions, with the highest level of exploration with reinforcement feedback, the lowest level of error feedback, and somewhere in between with both. This supports the idea that reinforcement feedback is driving this exploration, while error feedback is suppressing exploration. And when given in combination, they interact to do something unique. One of our goals is to simplify our model-based framework to minimize the number of assumptions that we're using. So I'll be skimming over a lot of our detail here, but please feel free to ask questions later. Our process starts with our general model, which we use to generate our a priori predictions. We then did a systematic reduction of parameters, generating 9 different models with new assumptions and fit them to our data using an AIC analysis. And from there, we were to select a single model that best encapsulated our data with a reduced parameter set. And using a bootstrapping procedure, we were able to find the 95% confidence intervals of the free parameters of this model. What's great about this whole process is that at the end of the study, we now have a new model to generate new predictions for new experiments. So here I'm just showing again our 36 individuals running through the experiment. And here I'm showing the results of our best fit model. And you see, even with a reduced parameter set, it still does very well in capturing the trends between each condition. Now, for our Parkinson's participants, we hypothesized that damage to the basal ganglia will cause a deficit to exploratory motor behavior. And specifically, we should see that people with Parkinson's will display lower exploration compared to neurologically intact age-matched controls. Here, I'm showing our lag 1 autocorrelation analysis for our age-matched controls. And here, I'm showing the lag 1 autocorrelation analysis for people with Parkinson's. And we found statistically significant differences between groups in the reinforcement condition, suggesting that damage to reinforcement-based neural circuitry is compromising task exploration. When we fit our best fit model to both groups individually, we see our model is able to capture the trends between groups simply by reducing the parameter associated with reinforcement-based processes. So in conclusion, our empirical results and model suggest that reinforcement boosts exploration while error feedback suppresses exploration. And when given simultaneously, they interact. By recruiting people with Parkinson's, we were able to gain causal insight into reinforcement-based processes and how they influence exploration. Specifically, that a compromised reinforcement-based neural circuitry reduces exploration. And to summarize our methods, we use a model-driven hypothesis to gain insight into how a particular process impacted by neurological disease influences and disrupts behavior. Now, the conference posed 3 questions to us. And for question one challenge that we see computational modeling can help address is gaining mechanistic insight into how a particular neural process influences sensory motor behavior and how that gets disrupted with neurological disease. For question 2, the way we used a model is to encapsulate our hypothesis and drive our predictions, which are then able to continually refine given more and more data. As such, one thing that we need to help support our modeling efforts is access to more data, either via large clinical populations with shared data sets or through collaborations with other labs. And one key opportunity we see with computational modeling is helping able to quantify disease onset and severity through personalization of modeling. And one challenge that we see is choosing the right model complexity that still encapsulates the behavior that you want to see while minimizing the number of assumptions needed to make that behavior. So with that, I'd like to thank you for your time, and I'd be happy to take any questions.
We have people with articles in both sides of the room, so we have time for, I think, one question.
So thank you so much for the interesting presentation. So I just wonder whether your model prediction about the Parkinson's disease is because Parkinson's specific kind of phenotyping, because Parkinson's disease stuff is kind of minimizing the movement. So it's a hypometria and then bradykinesia. So, and then, but in the same way, other basal ganglia disease like Huntington's disease that is more like a movement excessive. I just wonder whether your prediction is because you specifically chose a certain population. The same basal ganglia damaged population, their movement disorder kind of type would be totally opposite. Do you understand my question?
I'm sorry, I don't think I understand the question.
So I think that the reason why the reinforcement learning process based on the basal ganglia, that one could predict the Parkinson's disease well is because Parkinson's specific kind of characteristics. So
Mhm.
let's say that if you choose Huntington's disease
Mhm.
as a kind of reflection of the basal ganglia damage, then whether the result would be the same.
Mhm. I would hypothesize that we would see similar results to our Parkinson's disease. And I say that because although with Parkinson's, there is this reduced variability and slowness to initiate movements, and in Huntington's, there's this wide variability. We chose lag-1 autocorrelation analysis because it is a time series dependent analysis, not just the total spatial variability that a participant may show. And as such, even though they may exhibit different levels of variability, we still expect this sequential behavior to occur. Does that answer your question? Thank you.